Hello everyone, I am glad to have this opportunity to share our study published in Imeta, Probio-M9, a breast milk-originated probiotic, alleviates mastitis and enhances antibiotic efficacy, insights into the gut-mammary axis. Mastitis, a prevalent condition that affects nearly all lactating mammals. Mastitis leads to substantial economic losses worldwide due to the significant reduction in milk quality and quantity, directly impacting profitability and productivity. Meanwhile, a previous cross-species fecal microbiota transplantation experiment cows to mice found that intestinal flora imbalance may be a key factor in mastitis, and supplementation of probiotics to restore the function of intestinal flora may be an effective and safe strategy to treat mastitis. So, how do the bacteria in the gut and the mammary gland form a connection? Some relevant studies have found that during pregnancy, the gut will undergo a series of specific physiological changes, which promote the transfer of certain bacteria such as bifidobacteria and lactobacillus into breast milk through the gut-mammary pathway, and then directly to the baby through breast milk. Based on the above background, in this study, we evaluated the efficacy of the probiotic strain, Lacticaseibacillus rhamnosus Probio-M9, in treating mastitis by using a rat mammary model infected with Staphylococcus aureus, and the existence of a pathway of the gut-mammary translocation of Probio-M9. In Experiment 1, 72 female rats were randomly divided into 6 groups: positive control, blank control, antibiotic plus Probio-M9, Probio-M9 treatment, and Probio-M9 prophylaxis. After delivery, all groups, except for the blank control group, underwent Staphylococcus aureus infection via intramammary injection on day 0. Fecal samples were collected at 2 time points, 26 days ago and day 4. The blood samples were collected at day 4, and the mammary glands of rats that were fed Probio-M9 were collected at day 4. The results showed that the Probio-M9 could significantly inhibit the inflammatory response and restore the Th1, Th2 balance to normal level when used for the prevention or adjuvant treatment of mastitis in rats. This suggests that Probio-M9 has a positive effect in alleviating mastitis in rats. And then, with a further analysis of changes in the gut microbiome, Probio-M9 effectively relieved the lower alpha diversity caused by mastitis. And Probio-M9 increased the abundance of allostipes sp and decreased the abundance of Bacteroides nordi significantly. Moreover, administration of Probio-M9 significantly alleviated the alterations in the gene abundance of specific KEG and Kz. According to the correlation analysis, we found bacteria that were significantly positively correlated with differential KEG genes and interacted with immune cytokines. These results indicate that Probio-M9 can restore intestinal function and reduce inflammatory response in rats with mastitis by regulating intestinal flora. We conducted Experiment 2, which 2 key modifications based on Experiment 1. Firstly, we excluded antibiotic treatment from the experimental design. Secondly, we included 2 additional groups, a non-mitted group and a group of rats not challenged with Staphylococcus aureus, but administered Probio-M9. The result aided that both Staphylococcus aureus-induced mastitis and lactation were necessary prerequisites for bacterial translocation to occur, and bacterial translocation likely occurred through the lymphatic system. 85 cultures were isolated exclusively from and mesenteric lymph nodes of lactating rats. There were only 20 SNPs across all isolates and no common mutations were found between the original Probio-M9 strain and the 85 cultures. These results showed that these 85 isolates collected from mammary gland and mesenteric lymph nodes of the 12 rats were highly related to Probio-M9. Finally, this study is summarized. Probio-M9 has the potential to alleviate mastitis and mitigate the adverse effects of antibiotic therapy. Through genomic level analysis, we have identified instances of independent Probio-M9 translocation from the rat gut to the mammary gland, potentially involving the lymphatic system. Moreover, we have confirmed the requirement of lactation and the development of bacterial mastitis for bacterial translocation. While the precise mechanism of probiotic translocation remains unclear, our results suggest a selective rather than random process.